Hello students, welcome to padhobio. My name is Shashikant and in this video we will discuss about types of transport mechanism that occur across the plasma membrane. So as we all know that every cell has a plasma membrane which surrounds all its components. So what do you think? How does this plasma membrane regulates the movement in and out of the cell? Mainly phospholipid and proteins are responsible for these overall movements. So cell transport is basically divided into two types, that is passive transport and active transport. In passive transport, here there is no energy is required at all. This process is totally automatic. But in active process, it totally depends on the energy. So let's start from the first, that is passive transport. The first type of passive transport is passive diffusion. It includes simple diffusion, passive diffusion and osmosis. So what is diffusion? It is generally movement of anything that is from higher concentration to its lower concentration. Let's take an example of dissolving a cube of sugar in water. Slowly you will discover that the cube of sugar will dissolve evenly throughout the water. Diffusion occurs because of a concentration gradient. Difference in concentration gradient of two different areas, so sugar molecule moves from its higher concentration to its lower concentration. And this same diffusion occurs in the cell when the solute particles move across the plasma membrane from their higher concentration to the area of their low concentration. This totally depends on the difference in the concentration at the interstitial fluid and at the cytosol. Simple diffusion occurs in the non-polar molecules. Being non-polar, they can cross plasma membrane in between phospholipids, which make the plasma membrane, because this region is also non-polar in nature. Example of simple diffusion includes the movement of gases like oxygen, carbon dioxide or fatty acids. Here, as you can see, the concentration of oxygen is higher at the interstitial fluid, so it moves across the plasma membrane as long as the concentration gradient difference exists. If the concentration of carbon dioxide is higher at the cytosol, it will move across the plasma membrane towards the interstitial fluid. So this is all about the simple diffusion, where solute particles move from their higher concentration towards their lower concentration through a plasma membrane. And another type of diffusion is called facilitated diffusion. This method is used for small charged or polar solutes. Because of their polar nature, they cannot cross the plasma membrane, but they can cross the membrane protein with a process known as facilitated diffusion. This type of diffusion is also of two types, channel mediated diffusion and carrier mediated diffusion. These two methods depend on the type of proteins which is going to be used in this process. In channel mediated diffusion, here any molecule or ion move through a water-filled channel, and all protein channels are specific for specific ion or molecules. Also, there are two types of protein channels available in the plasma membrane, in which one is leaky channel. It is called so because it is continuously open, while another one is a gated ion channel. It has a gate which open for a stimulus. For example, if we talk about sodium ion channel, in sodium leaky channel, it moves continuously through this channel, while sodium gated channel only open due to a stimulus to allow sodium ion to pass through into it. On the other hand, carrier mediated diffusion includes the movement of molecules that are small, polar in nature, across the plasma membrane. Let's take an example of glucose molecule. Its transport is accomplished by a glucose carrier. During this transport, the carrier protein changes its shape to transport the glucose molecule from one side to another side. Now coming to another type of process, that is osmosis. It is also a passive movement of water through a selectively permeable membrane. This process can only be seen when difference in concentration of water molecule is present in any side of the membrane. To make up this difference, water molecule will move to the side containing its lower concentration, and this will happen in two ways. Water molecule can pass through plasma membrane or also through a protein embedded in the membrane known as aquaporin. You all know that plasma membrane is not permeable to most of the solutes. That is why it is known as semi-permeable membrane. Now let's take an example in which the interstitial fluid has more solutes than the cytosolic side. One side has 3% solute and 97% water, while the cytosol has 1% solute and 99% water. It means that more water content present in the cytosolic phase. So the water molecule will move down its concentration gradient from 99% higher concentration to 97% lower concentration to achieve an equilibrium through a plasma membrane. Now coming to active process. Here this process requires cellular energy for the transport process. These are of two types, active transport and passive transport. Here the movement of solute occurs against the concentration gradient when the solute moves from its lower concentration to the higher concentration. In another case of vesicular transport, it is a transport mechanism across the plasma membrane which includes vesicles. A vesicle is nothing but a membrane forming a sac-like structure containing some materials inside it. So active transport are also of two types, primary active transport or secondary active transport. Primary active transport, also known as direct active transport, it includes the cellular energy in the form of ATP, adenosine triphosphate, to transport solutes from their low concentration to higher concentration. It means against the concentration gradient using ion pumps or cellular pumps. Let's take an example. Here a pump is shown in the membrane, that is sodium-potassium pump. It requires 3 sodium and 1 ATP binding to start the pump to operate. ATP here used as a fuel to energize the pump in order to release the energy. ATP now breaks down into the ADP and phosphate. ADP is released and phosphate binds to the pump, which causes a change in the pump shape so that sodium ion released into the interstitial fluid, 2 potassium ion binds to the pump, and phosphate released into the cytosol. Also, after the potassium ion released into the cytosol, the pump reverts back to its original shape. In secondary active transport, it occurs when movement of any substance down its concentration gradient gives some energy for the movement of second substance against its concentration gradient. Let's say if a solute is moving down its concentration gradient, gives some energy to move another solute against its concentration gradient, that is low concentration to high concentration. Well, secondary active transport is also of two types. First is symport active transport and another is antiport. First is symport active transport, where two molecules move in the same direction, while second is antiport active transport. Here two molecules move in the opposite direction. Let's understand it with an example. Here sodium ion concentration is higher outside the cell and lower concentration inside the cell. It is a symport protein which creates an energy by the movement of sodium ion from higher concentration towards the lower concentration for the movement of glucose molecule from lower concentration to higher concentration. Now look at the antiport system. Here sodium and hydrogen concentration are higher outside the cell and lower concentration is inside the cell. So here this is an antiport protein. It allows the movement of sodium ions from higher concentration to lower concentration, which allow the movements of hydrogen ion against their concentration gradient. Always remember that movement in the same direction is symport system and in opposite direction at the antiport system. Now coming to vesicular transport. It includes the movement of larger substances into a sac-like structure known as vesicle. Example, movement of proteins or carbohydrates. It is also of two types, that is endocytosis and exocytosis. During exocytosis, the materials are exported outside the cells. This process starts in the Golgi apparatus, where a membrane is formed and all the materials are packed inside it, which are need to be transported. Vesicle gets fused with the membrane lipid bilayer and contents of vesicle are released outside the cell. On the other hand, in the endocytosis process, as the name suggests, it is opposite of exocytosis. Here the material which is need to be transported inside the cell is trapped in by the membrane, forming a vesicle in which the materials get packed. There are three types of endocytosis: phagocytosis, pinocytosis, and receptor mediated endocytosis. In the process of phagocytosis, the vesicle containing the material which is need to be transported inside the cell gets fused with the lysosome, which contain different digestive enzymes. The material which is trapped from outside the cell gets digested in the lysosome, and this process is also known as cell eating. Coming to another process known as pinocytosis, which is also known as cell drinking. It occurs when plasma membrane moves inward and engulfs some droplets from interstitial fluid that contains some dissolved solutes which are useful for the cell. Now the last process is receptor mediated endocytosis. This process requires some receptors which are present at the surface of the plasma membrane outside the cell and act as a signal. When a specific molecule binds to these receptors and the membrane folds, enclosing the membrane to form a vesicle. One important thing, endocytosis and exocytosis, both of these processes require energy. So that's all about the membrane transport. Hope you understand this concept, and if you like these type of videos, just go and subscribe padhobio channel. Take care and goodbye.